Hello, and welcome to the Meiosis chapter. We're going to be taking a look at this very special type of cell division process that occurs in only sexually reproducing organisms. And it only occurs in a certain part of the sexually reproducing organism's body. And that area or that organ is called the gonad. So this process, again, is a special kind of cell division, and the overall purpose is to produce egg or sperm, reproductive cells. And those reproductive cells combine to produce a brand new organism, an organism that is genetically unique. So, that said, let's take a look at the review topics. So some key terms. We'll take a look at some key terms in this meiosis process. And of course, the stages of meiosis. And what I'm going to focus on here in this review is the differences, major differences between mitosis and meiosis. And you're definitely going to want to understand those differences, those major differences. And we've already outlined a couple of them as far as the purpose of meiosis as opposed to mitosis. And then how meiosis produces genetic variation. We'll talk about a few mechanisms that are involved. So some terminology: a homologous pair of chromosomes. Every chromosome in your genome, we talked about genomes in the cell cycle video and in that chapter, has a match, a pair. So this chromosome here is shown already duplicated with its sister chromatid, and we have another chromosome that also is duplicated with its sister, and they have similar size, similar placement of the centromere, the same types of genes indicated here by these different colors located along the length of the chromosome. And the question is really, why do we have two of each? Where did we obtain each of these? Well, you obtained one from Mom. Thank you, Mom. And you obtained one from Dad. Thank you, Dad. So you have one of each. Two of each. One from each of your parents. Okay. So some more terminology. Somatic cells are the cells of your body. So, your liver cells, your skin cells, your muscle cells, all those are somatic. Now, the other types of cells in your body are the gametes. If you are a female, you have eggs, and if you are a male, you have sperm. The somatic cells all have two sets of chromosomes, just like we talked about. They have homologous pairs of chromosomes in the genome, and they have a diploid number. So their number of chromosomes is said to be diploid. So there are two of each chromosome. So N is the number of chromosomes in a single set. So you get a single set from one parent, and you have a set from each parent. So you have two sets, so 2N. So for instance, if I have, as in humans, you have 23 chromosomes that you get from Mom and 23 that you get from Dad. The number in a single set is 23. So 2 times 23, your diploid number is 46. You have 46 chromosomes in each of your body cells or somatic cells. Now the gametes have half as many chromosomes, and that makes complete sense. Why would you have half as many? Because the egg combines with the sperm to make that first diploid cell. And we call these cells haploid. They have half the number of chromosomes. Okay. And we call that 1N or just N because they just have one set. All right. So this just kind of reviews that idea of diploid. And this is an example from the PowerPoint. So 2N again, if Mom has 20 and Dad has 20, N would be 20, so your diploid number would be 40. Okay, so this is what you get from Mom, that's what you got from Dad. This is what you got in the egg, that's what you got from the sperm. Okay. Now another terminology is karyotype. It's just an ordered arrangement of all of the chromosomes. And nearly all of the chromosomes are something called autosomes. And then at the very end, the very smallest pair, well, not necessarily smallest, depending on the chromosomes that you inherit, are the sex chromosomes. So if you have an X and a Y, as shown here, you are a male. And if you have two X chromosomes, you are a female. Something I want to point out here is that the X is very large in comparison to the Y. That will be very important when we talk about sex-linked inheritance. Okay, so just keep that in mind for now. So another review, somatic versus gametes. Somatic cells are diploid, gametes are haploid, 2N versus 1N. And then we're looking at, how they are generated, when, where, why we generate these cells, these haploid cells. We're focusing in on this part of the puzzle. We took a look at this part of the puzzle back in the cell cycle chapter, talking about mitosis and generating just more diploid cells. Okay, so the overall process is shown here. And these two individuals were not dressed appropriately, so I took the liberties of dressing them up in these lovely costumes. But in any case, we have our haploid egg and our haploid sperm. And the haploid egg and the haploid sperm fuse during fertilization to create that first diploid cell, two sets, one from mom, one from dad in that first cell. That diploid cell undergoes many mitotic divisions and results in the multicellular organism. Each of these multicellular organisms can produce gametes. Females produce gametes in their ovaries. Males produce gametes in their testes. Those are the gonads. Okay. So I'm not going to, again, go through all the specifics as far as the different phases. You will need to know those specifics, but what I really want you to hone in on are the differences between mitosis and meiosis, and that's what I'm going to point out here. One huge difference occurs in prophase I. And in prophase of mitosis, the homologs have nothing to do with each other. They're there, but they have nothing to do with each other. They don't interact at all. In meiosis, it's completely the opposite. They pair up here in prophase I. The homologous chromosomes pair up, and that process of pairing up is called synapsis. And they form these tetrads, 1, 2, 3, 4, tetra. And they cross over, they physically exchange parts with one another. This is a huge source of genetic variation in populations. You're creating a whole new chromosome that has never existed before. Amazing. So the non-sister chromosome, so non-sister means chromosome of opposite homologs. So these two in red are sisters. These two in blue are sisters, but one here in red and one here in blue are non-sisters, okay? And those are always of the homologous pair. Those points of crossover are called chiasmata. Chiasmata, okay. Now we go through metaphase I. Again, another big difference here. The chromosomes are lining up together. The homologs line up together in pairs. They don't do that in mitosis. They do not line up together in mitosis. And that sets up a situation where in anaphase II, the homologs separate, whereas in mitosis, the sisters separate. Okay, so in anaphase I, the homologs separate. We go to telophase I. Okay, so now we go on to meiosis II, and in meiosis II, prophase II, not much different than mitosis. Not much going on there that's different. Metaphase II, we have two cells now. Okay, of course, because we've gone through that first round of cell division. Now we're going through the second round of cell division. So we see both cells lined up here with their chromosomes on the metaphase plate. And now the sisters finally separate here in anaphase II. So the sisters don't separate until anaphase II of meiosis. Another big difference. And another big difference here, meiosis versus mitosis, we have 4 cells. 4 cells form. 4 cells at the end. And they all have half the number of chromosomes as that original diploid cell from the gonad. But we start with a diploid cell in the gonad, and these cells are very special cells. They are destined to undergo meiosis, and we end up with 4 haploid cells. Okay, so the process is outlined for you in that chapter. We already talked about these differences here in prophase I. The homologs pair up, tetrads, synapsis, crossing over, chiasmata. Those are all important terms. And then, like we had said in metaphase I, the homologs pair up on the metaphase plate, and in anaphase I, the homologs separate and the sisters don't separate until anaphase II. Okay, and then finally, genetic variation. How do we produce genetic variation? The biggest source is crossing over, the biggest source. There are other sources, but that really accounts for nearly all, I have to say, nearly all of the genetic variation you see in sexually reproducing populations. This physical exchange of parts between the non-sisters. Now you also have something called independent assortment. That is how the chromosome pairs line up on the metaphase plate. We can result in different combinations here, so take a look at that. And then finally, just the chance that that egg fuses with that sperm. That's a 1 in 64 trillion chance. So you, my friend, are not just 1 in a million like your mom and dad tell you. You're 1 in 64 trillion. So you should feel really, really good about yourself after completing this video. So please, if you have any questions about this process, let me know, and I will assist you in whatever way I can. I hope this has helped you better understand meiosis.